Good morning, everybody, and welcome to my channel.
So today our first topic is the anatomy of cornea. Okay, so let's start. You all know this picture. So this is the very basic picture of our human eyeball, and in this picture, you all make out that this is the inner nervous coat, and this is the middle vascular coat, and this white color is the outer fibrous coat. Okay, so this outer fibrous coat is made of the cornea and sclera. So this cornea form the anterior 1/6 part of our eyeball. Okay, so this cornea is highly transparent structure and it is purely avascular. So what does this transparent and avascular words mean? The word transparent means the light can easily pass through it, and avascular means lack of any blood vessels. So our cornea does not have any type of blood vessels. Okay, and our cornea is also called as the watch glass like structure. Why so? As because the cornea is located at the very front surface, at the very front of our eyeball, just like a watch. We all have a wristwatch. So in that wristwatch there is a glass like structure at the top through which we are looking digit of this watch, isn't it? So in this slide you can see that. So this is our cornea and this is the internal structures of our eyeball. Okay, so this is iris and this is pupil and this above structure is the cornea. So that is the cause why this cornea is also called as the watch glass like structure. So in the next slide I will discuss about the some basic anatomical dimensions and property of this cornea. Okay, so what are the basic properties of the cornea? So our cornea, from this picture you can easily look out that our cornea has two surfaces. So this is the anterior surface and and this is the posterior surface. Okay, so our cornea has got two surfaces. So the front surface of the cornea is elliptical and convex. So elliptical means its horizontal diameter and vertical diameter are not equal, they are different. Okay, and convex surface means as like the convex lens. Okay, so in the front surface, the horizontal diameter of the cornea is 11.75 millimeter, and the vertical diameter of the cornea is 11 millimeter. Okay, the and the front surface refractive power is plus 48 diopter, as it is the convex surface. Okay, now come to the back surface. So back surface is circular, means its horizontal diameter and vertical diameter both are same, and this back surface of the cornea is the concave surface. Okay, so this horizontal diameter equals to the vertical diameter is about 11.5 millimeter, and the refractive power of this back surface of the cornea is minus 5 diopter. Okay, so till now we have learned that the front surface power of the cornea is plus 48 and the back surface power of the cornea is minus 5 diopter. Okay, so the total refractive power of the cornea is 48 plus minus 5, that is result in the plus 43 diopter. So the total refractive power of our cornea is the plus 43 diopter. Okay, and we know that the total refractive power of our eyeball is plus 60 diopter. So maximum refractive power is covered by our cornea. So cornea is an important refractive medium for our eyeball. Okay, and this refractive index of the cornea is 1.37. Okay, so now we are going to learn about the anatomical structures of the cornea. Okay, so there are six different layers present in the cornea anatomically from anterior to posterior, means from front to back. There are six layers. So these are, the first one is the epithelium, the second layer is the Bowman's layer, the third layer is the stroma, the fourth layer is the Duas layer, the fifth layer is the Descemet's membrane, and the sixth layer is the endothelium. The Duas layer is not included in this picture. This Duas layer is a corneal layer of recent origin, means it is discovered by the scientist Dr. Harvinder Dua in the year 2013. Okay, so after stroma there is Duas layer and then the Descemet's membrane and endothelium. So this is the very important picture of the anatomical structure of the cornea. So this is very important for your examination purpose also. Okay, now let's discuss about the epithelium and all the consecutive structures in detail. Okay, so the corneal epithelium. So the corneal epithelium means it is the first line of structure in our human eyeball. this epithelium is made up of stratified squamous type of epithelial. Okay, and the thickness of this epithelium layer is 50 to 90 micrometer. Don't forget this unit. This is not millimeter, this is 50 to 90 micrometer. Okay, and the epithelium of the cornea consists of 5 to 6 layers of cell, and there are 3 different types of cell. So the cell layer is 5 to 6 and cell is 3 different type. Okay, so don't forget this. Now, what are these different types of cells? So you know that this is the structures of the corneal layers, and if we look through a microscope, the magnifying image of this corneal epithelium is like this. So this corneal epithelium is consist of 3 different types of cells. Okay, so these 3 cells are, first one, the long cells. Okay, this type of cells are also known as the columnar cells. Okay, then this flattened cells, these are also known as the wing cells, and above the wing cells these cells are also flattened, these are also known as the superficial flattened cells. Okay, so this posterior most layers of the cell, the columnar cells, also called as the basal cell layer. Okay, so these are the polygonal cells which are located at the basement membrane of the corneal epithelium. Okay, and this layer forms the, or this layer is the germinative layer of the corneal epithelium. Why germinative layer? As because this cell layer undergoes the mitosis cell division and produces new daughter cells which are pushed anteriorly towards the wing cells, and this is the basis of regeneration of the epithelium. Okay, and the nucleus of these columnar cells is oval in size, and these columnar cells are attached to the other columnar cells medially and to the wing cells above through the tight intercellular junction. So the names of the intercellular junctions are desmosomes, zonula occludens, and macula occludens. Okay, so what is this intercellular junction? Just imagine, when we made a house, what we are doing, we not only arrange these bricks one after another, but in between the bricks we put cement. Okay, so it actually increase the integrity of a structure. So same to like that, this tight intercellular junction actually help to strengthen the structural integrity of the epithelium basement membrane, and as well as it acts as a barrier, and it is a resistance to the flow of water, electrolytes, any ions, any foreign body towards the cornea. Okay, so after these columnar cells there is the flattened cells. The flattened cells also called as the wing cells, because it looks like a wing. Okay, so these wing cells are actually the middle of these columnar cells and the superficially flattened cells. The nucleus of the wing cells is also flattened, and it is again attached to the columnar cells and superficial flattened cells with this tight intercellular junction. Okay, and next the superficial cells. The superficial cells, the nucleus of the superficial cells is again flattened nucleus, and the superficial cells also linked to each other with this tight intercellular junction, and anterior surface of the superficial flattened cells is covered with the microvilli. So each microvilli is of 0.5 millimeter in height. So what is the significance of the microvilli? This microvilli increase the stability of the precorneal tear film. Okay, so this is all about this anatomical and histological structure of the corneal epithelium. So what is the function? So as we have learned that this epithelium is the anterior most layer of the cornea, so it actually protects the internal structure of the cornea, and it also acts as a barrier to the flow of barrier and resistance to the flow of water and electrolytes, foreign particle from entering into our cornea. Okay, so let's discuss the number 2 layers, that is the Bowman's membrane. So we have completed this layer, the epithelium layer, and then after the epithelium, the very narrow layer, this is the Bowman's layer or Bowman's membrane. Okay, so this Bowman's membrane, the thickness of the Bowman's membrane is 8 to 14 micrometer. 8 to 14 micrometer. Okay, and the most interesting fact about this Bowman's membrane is that it is the acellular membrane. Okay, so this acellular membrane means this layer of the cornea does not have any cells. So as there is no cells, there is no mitosis, and this is the basis for the cause of not regenerating the Bowman's membrane. means when there is any injury or any trauma due to which there is loss of the epithelium and loss of the Bowman's membrane. Suppose the epithelium can regenerate as the columnar cells of the epithelium can mitosis and make the new cells, but this Bowman's membrane has no cells, so there is no mitosis, so there is no regeneration of the Bowman's membrane once it is destroyed. Okay, and this Bowman's membrane is not a true membrane also, it is the superficially condensed part of the its underlying structure, the stroma. Okay, so what is the function of Bowman's membrane then? The Bowman's membrane actually connects the underlying stroma with the basement membrane of the corneal epithelium. Okay, so now comes to the stroma. So the stroma is the thickest layer of the cornea and it constitutes the 90% of the total corneal thickness. The thickness of the stroma is 0.5 millimeter. So it is the thickest layer. Okay. So what the stroma contains? The stroma contains three different things. The first one is the collagen fibrils. So these and these are the collagen fibrils. Okay, and then the cells, different types of cells like keratocytes, like macrophages, lymphocytes, different types of cells, and the ground substances. Okay, and the name of the ground substances is the gags. So what is actually look like? Okay, so if you consider these ground substances as water, and all these collagen fibrils and these all the cells are actually soluble, are embedded in these ground substances. Okay, now you can understand, I think. Let us discuss in detail about this collagen fibrils. Okay, so the collagen fibrils are many in number. Okay, and this collagen fibrils is arranged in a particular manner within the corneal stroma. It is not arranged or it is not located haphazardly. There is the peculiar arrangement of this corneal lamellae or collagen fibrils. Okay, so what is that peculiar arrangement? These corneal lamellae or these collagen fibrils are parallel to each other, and at the same time they are parallel to the corneal plane also. So this is the peculiar arrangement. See this picture. So let's consider these are the collagen fibrils. Okay, so it is the first line arrangement, 1 2 3. Then in the second line, in the second line, the collagen fibrils are located in the gap of the first line. Then again third line it is same as the line 1, and then in the fourth line it is the same as line 2, like this way. Okay, so what is the significance of this peculiar arrangement? Means if there is means if the lamellae or fibrils are arranged haphazardly, so what happens? So the clinical significance of the peculiar arrangement of the corneal lamellae is to maintain the corneal transparency. Okay, so if these are arranged haphazardly here and there, the cornea is not at all a transparent structure. Okay, so the peculiar arrangement is one of the unique factor or unique properties of the collagen fibrils in the stroma. Okay, so and we have already told that there are different cells and which are actually embedded or soluble in these ground substances. So what are the cells? The keratocyte cells. You have seen that these are the keratocyte cells. Okay, so this is the nucleus of the keratocyte cells, and there are different processes, elongated processes of these keratocyte cells. Okay, and this keratocyte cells is 2 to 4 millimeter long, and these keratocyte cells actually producing these collagen fibrils during the embryogenesis and when it is injured. So the new collagen fibrils are produced from these keratocyte cells, and apart from these keratocytes there are also other cells like wandering macrophages, lymphocytes. So these are actually the immune cells. So but the question is, these immune cells, means this white blood cell, should present at the blood vessel, and we have stated that the cornea does not have any blood vessel. So from where this actually comes? So in our next part, when we will study about the blood supply of the cornea, we will study that this cornea is purely avascular, but in the extreme periphery of the cornea up to 1 millimeter there is presence of a few blood vessels which are also called as the pericorneal capillaries or pericorneal blood vessels. So from that blood vessels the immune cells, the lymphocytes, wandering macrophages are come to the corneal stroma. Okay, so this is all about today. From our next lecture we will continue from this Duas layer and end the anatomy of the cornea. So for today I will end my lecture here. Okay, so thank you everybody.